Okay, it's 9:30. Can you hear me?
Yes. Yes.
Okay. I will share my screen to start with the webinar. While people are still joining, can you see the screen?
Yes.
Okay. Okay, I think I will start since it's already 9:30. While people are still joining, I'm going to give a brief introduction for the webinar. First of all, welcome to EU-LI-PHE Talks, and thank you for being here. Now, this is an initiative webinar series organized by the European Network on Livestock Phenomics Cost Action, the EU-LI-PHE Cost Action. Now, in this first episode, we are going to give an introduction to livestock phenomics. Now, before entering the specific webinar agenda, well, I'm going to introduce myself. I'm Agostina Zubiri Gaitan. I'm part of the Young Researchers and Innovators group. And well, first of all, I would like to thank the whole group for their help in the organization of this series of events. And well, this is a photo of a meeting that we had this year in Florence when we got involved in the organization of this event and many others. I would like to especially thank the Young Researchers Committee. I think we will be able to introduce ourselves probably later. And I would like to thank Luca Fontanesi, the chair, and Thomas Norton, the vice chair of the Cost Action, for giving us the opportunity to get involved in the organization of these events. Now, to start, this webinar will have two talks. The first talk will be given by Luca Fontanesi, and he will talk to us about what is phenomics. Now, Luca is a full professor in animal breeding and genetics at the University of Bologna. His research interests are mainly towards genomics, metagenomics, metabolomics, and phenomics in general, and their application in the animal production sector. And as I already mentioned, he is the chair of the EU-LI-PHE Cost Action. Now, the second talk will be given by Thomas Klingström, and he will talk to us about phenotyping technologies used in livestock species. Thomas is an engineer in molecular biotechnology and a doctor in bioinformatics. He works at the Swedish University of Agricultural Sciences in SLU. And he is currently the coordinator of GigaCow, which is an initiative of SLU, which has the aim to build an infrastructure for the digitalization of dairy data collection to facilitate the exchange between researchers and farmers in the area of precision dairy farming. I would like to thank you both for being here and for agreeing to talk to us about these subjects. And after these two talks, we are going to have an interactive Q&A session, and where I invite you to please ask all the questions you need, to share your experience, so we can talk all together about this phenomics new area. And before starting, I will also like to ask you to join us. Now, in the QR code of the upper right side, you can scan it to go to the EU-LI-PHE webpage. You can read about the aim of the Cost Action and of the different working groups, and you can join us. And in the QR code of the bottom left side, you can find the links to all the social media platforms that we have. And I invite you also to follow us in those platforms where we publish all the events that we have and all relevant information regarding the construction. And I think I will put again this slide at the end of the two talks, so you can scan them in case you haven't done it already. And now I give the space to Luca to start with his first presentation.
Okay, very good. Thank you, Agostina. Can you see my screen?
Yes.
All right, very good. Okay, thank you, Agostina, for the very good introduction. And for me, it's really an honor to start this webinar talks series on Livestock Phenomics. That will be further developed during the next month. So stay tuned. This is a final message. For me, it's quite challenging because I need to introduce the concept of phenomics. What is phenomics? And to start with, we probably need to go back to some interesting backgrounds and why we need some improvements in the phenotyping approaches, especially in animals, but not only in animals, but also in plants, in humans, and so on. In particular, in animals, phenotyping is increasingly being recognized as a limiting factor in all application of livestock genetics and genomics. So the first area of application of phenomics is in animal breeding in some way, but it is not the only one. And probably, I don't know if it is the most important, but it is paired with the other main area in which phenomics is needed. Phenotyping is fundamental to routine and daily management of livestock populations in order to optimize reproduction strategies, disease control, and welfare of the animals. That means precision livestock farming in some way. And we need actually new phenotypes to foster these two areas, new ways to measure phenotypes, and new ways to use these phenotypes for these two main applications. So the phenomics is emerging as a whole, as a major new technical discipline in biology. Phenomics, the terms phenomics is paired to the omics approaches, in which there is genomics, transcriptomics, metabolomics, and so on. And probably will be one of the most important disciplines to produce big data in the future. We can define phenomics as the ensemble of methodologies and technologies for the acquisition, analysis, and exploitation of high-dimensional phenotypic data on an organism-wide scale. So what does it mean, organism-wide scale? We can also define animal phenome as a sort of ensemble of the information that can construct an animal. It is considered the physical and molecular traits of the animal. So we can identify many types of phenotypes at different levels that can in some way describe and dissect the animal phenome at many different levels. We can now try to classify the different types of phenotypes. And animal phenotypes can be classified in many different ways, according to the level in which they are measured, cell, tissue, organ, and whole organism, according to the type of information that is recorded, how we can measure, what type of information can be recorded. The temporal acquisition of the information, it is a single event, is a continuous event, is a combination of events, and the objective of the collected parameters. So the first level of complexity can be easily identified by the way in which we can classify phenotypes in animals. Then there is also another level of classification. We can target phenotypes in some way. Phenotypes can be dynamic, can change rapidly within short periods of time in the animals, and which is the time point, the most important time point in which we can get the information, or they can be stable. That means minimal change across a predetermined time window. For example, stature of the animals, of course, can change, but not in a short time period. Another level of classification of the phenotypes depend on the way in which we see the animals from which point of view. The phenotype can be external or final phenotypes. There are usually the most relevant phenotypes that have economic value, performance, morphological traits, disease resistance, behavioral traits. They are final phenotypes that give economic value to the animals in some way, or they are the objective of the breeding schemes, for example, of breeding programs, or they are internal or molecular phenotypes. We can call them endophenotypes. They are, for example, the level of the presence or absence of different types of biomolecules in the blood, in many other biofluids and tissue and so on. So this is a quite interesting way in which we can identify phenotypes. The final phenotypes, the external phenotypes, are determined by the contribution and interplay of many molecular phenotypes with the multi-level relationships and their interaction with environmental factors. So we cannot skip the environmental factors, and we need to manage these different levels. So this is the overview of how the different level of information can interplay between each other. So we have a genetic perturbation, genome level with diversity. We then can collect information from many molecular phenotypes, internal phenotypes, and they construct external phenotypes. But of course, there are environmental perturbations that modify and interplay between these levels. Do you know other way in which phenotypes can be classified? Please get in touch with us, because we would like to just try to summarize in the best way phenotyping approaches, phenotyping technologies in animals, because we also try to compile a Livestock Phenomics textbook. So we need also some advice from you. So there is clearly a broad heterogeneity in phenotypic classes. That depends from the level in which phenotypes can be collected, recorded, measured, but also from the heterogeneity of the animal species that we deal with. Livestock species go from large animals to very small animals, with many different objectives in terms of breeding. What is the purpose of the breeding? Where the animals are raised, how they are raised, why they are raised. So there is a wide array of scientific approaches and technologies that can be used to capture and manage phenotypic information. This is clear. There is a quite large complexity in this context. So why we have a class action in this field? Because we want to answer the main challenges that Livestock Phenomics can give us. We want to foster the development, integration, organization, and practical implementation of technologies that can be used to measure phenotypes, tools, methods, approaches, and models, and also expertise and resources useful to scan and interpret the animal phenomena. We need a lot of expertise. It is not obvious. Or just one scientist cannot cover the complexity of Livestock Phenomics, as I just try to summarize. And we want to pave the way for novel scientific knowledge and application in the livestock production sectors. This is the aim of EU-LI-PHE, the cost action that I'm sharing. And of course, don't forget to join us. Additional hints will be given later after the two presentation. And thank you for being here and listening. And I will be, of course, open for any question later after the second presentation. Thank you.
Thank you very much, Luca, for your short and very clear presentation. And we will now start with Thomas' presentation. Thomas, if you could share your screen, please. You are now muted.
Yes, I know. I need to, let's see here now. I need to get the request approved to share it.
You are now, I think, wait, I put you a co-host. So now it should work.
Perfect. Yes. Yes, so my core work is in dairy research, but and my background is I started up as a molecular biotechnician, which means that I find it very frustrating when you have more than one cell.
Excuse me, Thomas, you're not sharing your screen.
Okay, so I was not approved yet when I see. So now?
Yeah, now we can see it. Thank you.
Yeah, so my background is molecular biotechnology, which means that I was very much founded in an approach where we looked for one gene, one trait. And then I got the opportunity to move from biobanking and large prospective cohorts to working with dairy cattle, which means that we get a much more holistic approach to measuring all the characteristics of an animal. And what we're seeing now is that with the amount of technology that is coming into dairy farming, we need to actually start having people who are experts on the technology and the establishment of data sharing networks. Because in our case, for example, we have 15 different farms contracted to supply data for dairy data research. And then we also have 3 research farms owned by the Swedish University of Agricultural Sciences. You see on the map to the right, you see a bit of a population density map of Sweden mixed in with other factors. So the blue dots are where we have the dairy farms that are collaborating in the data collection network. And working on all this, I got a very fundamental question, and that is, we have a lot of geneticists, but I've never met someone who called themselves a pheneticist. And if we think about it from 100 years ago, then a genotype and a phenotype was both very, very theoretical concepts, because we knew that we had the concept of genes that were the basis of inheritance, but no one had observed DNA or understood how this information was transmitted between generations. And likewise, the phenotype, so we had this concept of so Willem Johansen introduced the concept of a genotype, meaning all these inheritable factors that you could transmit from generation to generation. That was the genotype. And the phenotype is the set of observable characteristics or traits of an organism. But even back then, when the genotype was a theoretical concept, they had, by this time, using applied statistics and inbreed and very homozygous gene plants, they had been able to develop the concept that there really is something like a gene which we inherit. And what made Willem Johansen special was the fact that he could show that even if, for example, 2 peas had the same genotype, they would have differences in characteristics in terms of size, color, etc. Which is because when peas are growing inside the pod, even if they're identical to each other, they will have different amounts of nutrition available to them based on their position in the pod. So that was his research that really showed that there is something about the relationship between the genotype and the environment, that means that we have a variable set of traits that we can observe from different individuals with the same genotype. And I think this has created a bit of a problem when it comes to phenotyping and how we think about phenotypes, because genetics as a subject has a very strong core, pretty much like if we think about onions or the earth, you have different layers. And in the core, when we talk about epistemology and ideas, it's often very quick to talk about things on a very theoretical level. And then as we move towards different levels of application, it becomes much, much more nuanced and heterogeneous. And in genetics, we have had this core concept of a gene to talk about. And then that means that people are geneticists. And I would say that perhaps the gene as a concept has been so strong that it's now holding us back in how we think about genetics. Because we know that for diseases, it's quite often very efficient to look for a single gene that is causing some problem. But we see, for example, with Alzheimer's, we do know that there is one specific gene related to early onset Alzheimer's, meaning that if you're homozygous for that gene, for that allele, what happens is that you're going to have a very, very high risk of getting Alzheimer's in your 50s. But then for most people, the risk of Alzheimer's is a statistical concept where we don't know if we're going to get it until we get it. But some people have a higher risk of getting it. And we can, pretty much like we can nowadays estimate the likely yearly milk yield of a cow, we can do that with Alzheimer's as well, but it's a binary outcome. But when we work with phenotyping and phenomics, we don't have that strong core concept because, as Luca exemplified, we have many different ways of measuring traits, the traits that make up the phenotype. And we also have many different kinds of traits that we try to measure. So becoming a pheneticist almost always boils down to the fact that we're not working on phenomics as a research subject at the core, where everything is more like homogeneous and abstract. What we're doing is that we're working at the surface level, where people, depending on what we're working on a specific set of traits within the entire phenotype. And this becomes quite hard when you try to write an article like we did that I will present, because just like if we look at Earth, for example, if we try to describe every kind of building on the surface of Earth, it will take an almost infinite amount of time because there's so many different ways we can build things. But if we cut it down to the mathematical modeling, we can say things like, if you're trying to build a residential building, using the normal soil types on Earth, we can have a ground pressure of roughly 750 kilograms per square meter. While if we want to build a factory or an apartment building, we need to look for solutions that allows us to have a pressure of 1,500 kilograms per square meter. So this is what we tried to do in this paper that I'm going to talk based on. Because what we had as the first question was, what technologies are available for low-income countries when it comes to high-throughput phenotyping? And working on the article, I realized that the theoretical basis for phenotyping makes it very difficult to give a comprehensive overview. So instead, it becomes more like a survey of different ways of thinking about phenotyping. And I think that is also the best way for the audience to think about this, that instead of thinking about the phenotyping as one subject, we need to think about which part of the phenotyping toolbox should I be using within my line of work, and how can we develop the theoretical framework for that component. So also looking back at having very similar characteristics to thinking about phenotyping as Luca, that we have different kinds of measurements. Like in Sweden for dairy cattle, I would say that the most classical phenotype to think about is just the color of the animal, because we have the Swedish red and white cattle, which is this aggregate mix of indigenous breeds that were red, and we started breeding on roughly 120 years ago. And then we had something called Swedish lowland cattle, who, because of the fact that they were black, we started to mix them with Holstein. And because of the massive success of Holstein breeding in Sweden, almost all of the genetic basis of the Swedish so-called lowland cattle was outcompeted by Holstein, meaning that a black Swedish cattle today got a completely different genetic background than it had 120 years ago when we started with systematic breeding. But at the same time, one-time measurements can often seem trivial. But when I was, for example, listening in to a dairy farm in the GigaCow network, where they were selecting bulls for insemination, and I would say that the two most important traits that they were selecting bulls on was, one, the size of the bull, because we have a problem that larger animals tend to be more productive. And that means that many barns today are undersized compared to the size of the animals. So farmers are willing to sacrifice milk yield for having smaller animals. And the other really important trait for them was the teat placement, which we know it is it's a very quantitative trait, meaning that, yeah, the infinitesimal genetic model works best to describe it. And what have happened is that the teats have gotten too tight on the udder, so they really don't want to inseminate their cows with semen from bulls that have offspring that get this tighter phenotype. And then we have the longitudinal data, which is an area where dairy cattle and also dairy sheep and dairy goats fit very well. Because with these animals that have continuous production, we can measure it on a daily basis. In this case, what you see here is the dots are the daily milk yield of a dairy cow between the 1st of January 2022 and the 2nd of November 2023. And what you can see here is that we have the end of the cow's first lactation, because this is when she got into the SLU dairy data collection infrastructure. So I have the milk yield from the second part of the first lactation. Then you see that you had the dry-off period. And then we have a complete lactation from this dairy cow. And then another dry-off period. And now she is in her second lactation, or third lactation, I mean. And we monitor this over time. And of course, this is one of the challenges when we're doing longitudinal data. It takes a lot of time to accumulate the data. It would be a benefit to work, for example, with broiler chickens who only live for 1 to 1.5 months, because then we can capture the entire life cycle. On the other hand, a broiler chicken, we only get the output data at the end of their lifetime. And we can also here see the importance of combining different measures. The red line you see here, that is the temperature humidity index at the farm during this period of life of the dairy cow. So what we see here is that it's very hard to get the information because you have such high variability. And one of the things I would really like to do is to get more collaboration with specialists in signals processing and regulatory theory, because we need people who can interpret repeated measurements as a kind of signal, and from these signals define a trait in the phenotype of an animal. So I think that in order to become better phyneticists, we need to look more and more into regulatory systems and different kinds of modeling. And then, of course, we have, as Lucas spoke about as well, the behavioral measurements. I would say that this is an area where AI and automated image processing is going to become incredibly valuable. Because if we work with, for example, a wearable device, we get these like nice data points, like, for example, an accelerometer. If an animal is economically valuable enough to be fitted with an accelerometer, we can calculate the movements of the animal using the accelerometer and then define a large number of different traits from the same animal. But we can get a much more holistic image of the animal if we, instead of a wearable device, can mount cameras and measure the behavior using the image processing technology. And this is also one of the things that we concluded, that we're going to see a huge change now with image processing, because if you mount a camera in a farm, you don't need to put the wearable device on the animal, which helps us work with animals that have a lower economic value for the farmer, because it can spur investments. So it becomes a lot of interplay between data science to process data, farmers and what value they can see from measuring more things on animals, and us researchers in being able to interpret it. And then we have the concept of endophenotypes. I have a lot of colleagues who are a bit skeptical because they think that functional traits are more closely related to what we want. But one of my favorite examples is actually already from the 1970s, because what they did was that they implemented heart rate monitors on a number of chickens. And then what they were doing was that they selected chickens in order to select for animals that displayed lower levels of stress. And then when they implemented these heart rate monitors, what they realized was that the animals that displayed less stress behavior, they were actually having higher heart rates and displayed higher levels of, internal stress, which is likely because they were selected against doing the things that normally made them calm down. So thinking about this, that, yeah, we can measure a trait that we think is relevant, but it requires a lot of validation to know that it actually works. And then lastly, something that I think that is also becoming much, much easier now is contextual phenotyping, meaning that we can measure the environment around an animal and then we track the response, like we are doing now with heat stress and the temperature humidity index, because it is actually now in the European Union, we have the Copernicus program, which launches different satellites, like the Sentinel-2 satellites that measures different bands of light. And from this, we can do things like deducing the amount of biomass on a field. And what we can, as researchers for phenotyping, we can let the animals out on the field and see which ones grow the best with different levels of biomass, as long as we know what plants are growing on that field. And these like high throughput measurements where you combine something high tech that is very generalizable, like satellite images, and then you can have very basic measurements such as weight or milk yield. That is a big game changer for us. And what it means for us, especially for young researchers, is that if you develop your toolbox for phenotyping, you can contribute a lot of value to many different projects with spending a little time in each project, but enabling very large and high impactful research. So that is something to think about when planning your future career, that you may see yourself as a geneticist, but the toolbox that you develop for measurements can be applied for a large number of different traits. And I think that is like the biggest take-home message you can make as a young researcher thinking about the future of research. And this is one of my favorite papers that we were citing in the article as well, thinking about what different levels of technique are we working on? because the most basic level is that you have a sensor and you make raw measurements. But a raw measurement can be quite meaningless. Like, for example, we're now working on a methane research project where we measure the electrical conductivity in the air right next to the mouth of the cow when she's eating while attending a milking robot. And what that does is that it gives us a measurement on how much methane she burps out, because 85% of the methane generated from a cow comes out of the mouth. So what happens is that when the cow burps, we get this indirect measurement of how much methane she emits. But then you have to spend a lot of time developing the data algorithms necessary to interpret this as a trait for methane production. So that means that level one results are quite limited in their utility. But when we combine it to the next step, then we can really start, when we start doing these detection algorithms, that is when we can start measuring advanced traits. And then by integrating it with further levels of information, we can get the real actionable results that generates value to the farmers as well as to the researcher. And if we think about, okay, so we have a lot of different traits that we can in theory measure, then the question is, of course, what is actually measured? And I've been using the Nordic Total Merit Index as an example in the article. So the Nordic Total Merit Index is something that has been under development for 100 years, pretty much. And what it is the index that defines which animals are the most economically valuable for the farmers. So 1 index point is equivalent to €1 more in income for the farmer. Or rather, €1 more in earnings. Because some traits, like for example, milk traits that I'm using here, they are of course associated with higher income for a farmer. But you can also do things like improved claw health. So you know that if you reduce the rate of fungal diseases on claws, on the hoof of the cow, then you can save, like for example, then you can save 10% of the claw trimmings, and that in turn means that you might be able to save €30 or something. And then what we need to think about is, how do we connect these merit traits? Like for example, we do the measurements. So weighing milk is simple. Actually measuring the amount of protein and fat in the milk is absolutely vital, because otherwise you will be selecting in favor of more water in the milk, because that's the easiest way to increase the weight of the milk. But by adding the chemical analysis, then you can actually get the trait that you desire. And what we did, and then we also looked at which traits that are used in the Nordic Total Merit Index also have a scientific definition in the ontologies available to us animal researchers. And if we look at it, what we see is that most of the traits used, even in a quite industrialized region like the Nordic countries, what we see is that most of the traits that are measured for the Total Merit Index, they're done with recordings, meaning that someone is looking at the animal and writing things down. Then you also have evaluations where you have an expert, where you, for example, evaluate the body conformation or the temperament of the animal, which requires a lot more training, but it's still something that's written down. And I think that the biggest help that we have developed in technology over the last 100 years is that the fact that we nowadays have mobile phones, which means that we can transfer the data back to centralized records, and we can also have this active exchange between the farmers and on-farm staff that make the recordings and the central databases, which can give us these actionable insights by integrating information in level 2 and 3 by that previous paper with Henkemann. And then we have a few high-tech ways of analyzing things like the chemical analysis. But even in a heavily industrialized country, most of the traits we track is done by pen and paper at the first, and then transmitted using mobile phones. And if we think about it, economy defined a lot of things of what we can do. And one of the questions we got while writing the paper was, why are you so focused on cows? Which was, of course, partially because I am working at the dairy data collection infrastructure. But at the same time, the reason that I'm working at a dairy data collection infrastructure is that we generate a lot more measurements in the dairy sector than for example, pigs and chickens. And what this illustrates on the turquoise charts, they are the data from the UN-FAO on how much value one animal generates during 1 year. And on the right side, we subsetted it to only look at the EU members. And we did this because for most animals, the EU data is much more homogeneous in terms of economic value because we have a single internal market. And what is worth noting here is that dairy milk, as a European dairy cow, is worth somewhere around 4,500 euros per year, meaning that a dairy farmer can invest quite a lot of money in milking robots, activity sensors, and those kinds of things, which has driven our ability to phenotype dairy cattle. If you look at instead at, for example, a chicken, then a chicken used for meat production, she's going to be worth somewhere around 4 euro, which means that you're never going to be able to pay from an industrial perspective to develop sensors that allows you to track chickens with wearable devices. But what we do have instead is now, and what we see on the industry side is that huge investments are coming in using audio and image analysis to track the behavior of, for example, pigs and chickens. Because if you can mount the equipment inside the farm and then keep track of many animals at the same time, then the cost of phenotyping a single animal becomes much lower. And what we're working on towards nowadays is to integrate data so that instead of doing a single measurement, for example, milk production, what we want to be able to do is to separate different events in the life of the cow. Like, for example, if you have a cow that have a high genetic potential for milk production, and it does not live up to that milk production, we don't, at the moment, it's very impossible to tell, was the genomic prediction wrong, which it is quite often. It's just correct often enough to be useful for breeding. Or were there a number of adverse life events for that cow that means that she never lived up to her genetic potential for milk production? Heat stress can, for example, reduce the milk production quite a lot in the summer. If she has subclinical mastitis, she's also going to have lower milk production. And being able to disentangle different traits within the phenotype of an individual, that is what we can really start working on now with the automation of agriculture and the implementation of machine learning algorithms, new wearable devices, better data processing techniques. When we have that, we can start thinking about not only measuring a single trait, but adding up the information so that a composite trait suddenly can be divided into a number of subtraits, which we can correlate with the genome of an individual. And that's how we get into this proper functional annotation of animal genomes. And what we're doing in SLU GigaCow is that we try to combine as many different projects as possible so that we can get those results that we really want. And I like to say that 5 years from now, we want the research to be useful for dairy farmers. And 15 years from now, we want the information to be useful for genetics research so that we can properly understand not only the single genes that are kind of easy to interpret, but also being able to understand the important contextual information that is created by the structure of the genome and interaction between different regulatory elements, because we thought that the genome would be like reading a book when we started the Hugo project. But I think that the genome contains a lot of sarcasm, in the sense that you need contextual information to know what something actually means when we read the genome. And something also worth keeping in mind is that we think about all the measurements from an animal perspective, but an animal is always living within their environment. So that means that a lot of the data we collect is generated on the farm or in the living environment of the animal. And then we need to figure out ways to integrate this information into the timeline of a single individual so that we can phenotype them properly. And on that note, I will end this presentation and open up for questions.
Thank you very much, Thomas, for your presentation. It was very interesting. So I think now we can start with the interactive discussion of these two presentations of phenomics in general. So I will ask you if you have any question, if you have any comment, if you want to say something, to please raise your hand or just unmute and talk. And I don't know if there's a way to raise the hand, but you can just unmute your microphone and start asking questions. So, well, I can start with something. Yeah, sorry, Tim. Okay, Pekka, right?
Yes.
You can.
Yeah, good morning. Pekka Oimari from University of Helsinki. I have a question like as Thomas was saying, many of these traits are measured now by human, either eyeballing or some scale, like from 1 to 5 or 1 to 10 or 0 to 10. In this cost action, how much do you pay attention for these traits that to be able to develop other ways of to get more objective or more precise measurement for those traits that are currently used in genetic evaluations, but the measurement technique may not be the optimal one? Thank you.
Sorry, your question was for, can you?
Did you hear me, and did you get the question?
Yeah, I think if you can repeat it, please.
Okay, so maybe Luca or Thomas can answer this. So my question was that, as Thomas was saying, currently in these national genetic evaluations, we have plenty of traits that are measured by eyeballing or in a subjective way, and then the others are from like in a scale 1 to 10, and again done by personnel or other less trustable methods. And I was wondering if there's aim during this cost action to pay attention for these traits, how to get more reliable measurements and more objective measurements for these existing traits, not only creating the new traits, new phenotyping. That was my question. Thank you.
Okay. Yeah, I can try just to answer. Then Thomas, you can add, of course, because probably you are more expert than me, especially for cattle, where these systems can work quite intensively. Phenomics means we want to improve the collection of the information from the animals. That means the use of new technologies, including images, can potentially substitute the subjective measurement. Of course, what is the bottleneck in all technology applications is the cost of the system. And if it is economically sustainable to add additional technologies, yes, of course, the process will smoothly go in this direction. And of course, there is the need to develop approaches, tools, methods to measure, to actually analyze the data after all, because if you collect a lot of data and the collection is so cheap, but the process of the data is complicated, there is another bottleneck there. So in the construction, we are working to just collect the relevant information technologies and data in order to facilitate this transition process, because it is clear that there will be a transition process that will go in this direction. The collection, automatic collection of all information from the animals in the future will be something that will be probably a quite routine process, including additional technologies that at present only a few farms can afford, on just a few species can afford, because the economic value can give the possibility to implement these technologies. So there is a obvious process and a transition process that will go into this direction.
Yeah, for me, I would say that, for me, the frustration has always been that phenotyping is not really treated as a research subject. It's just something that we do. We're not like in bioinformatics, for example. Actually, at my university, the Swedish University of Agricultural Sciences, we have actually had a lot of opposition against the concept of bioinformatics as a research subject. It took a lot of work for us to get it approved as an area where you can get your PhD. It was actually me who managed to put that through. But what happened is that, I would say that we're still at the start in the process of working on community level recognized measurement ways of transforming measurements into actually scientifically defined traits. So I think the cost action this has always been actually why I've been excited and supporting Luca in getting this cost action, which was quite a lot of work for him, I know. And so I'm really, really happy about this, because it creates an environment for us to start talking about how we can convert measurements into actual traits, and then incorporating these different traits into a phenotype that makes sense. And my two big role models when working with this, one is ICAR, which is the International Committee of Animal Recording, who are quite dominated by dairy cattle, but it's not the only thing they do. So they have this handbooks on things like how to define the milk yield of a cow, because since we decide when a cow goes on dry off, there is not like one objective way of saying how much a cow produce. We need to take into account the management decisions as well. And then the other thing that I think is a really, really interesting resource to look at is the phenotype knowledge base, which I put a link in the chat. There's one person who has difficulty reaching it. It works for me, so I think it should work for most people. But what the phenotype knowledge base is a database where they define algorithms for getting data from electronic recording systems from healthcare and turning that into phenotype. phenotypes for people. So it's like, for example, how do you go through electronic journals to define which patients got diabetes type 2, for example? So that means that you have inclusion criteria of which individuals do you evaluate, what diagnoses on the long list of potential diagnoses and treatments does actually mean diabetes type 2, and then how do you model, like, for example, how much response an individual has to different levels of insulin. So it's a really interesting concept on how to work with phenotyping from electronic records. And if there's something we have now in agriculture, it's electronic records.
Thank you both. Thank you for your question, Pekka. Thank you both for your answers. Does anyone else has a comment? Do you want to ask something? Now,
Can add,
about
yeah, well, go
sorry.
ahead, Gina. No, just a comment. The construction is an environment, is a network in which there will be discussions on many topics related to that. And of course, we support the networking activities and the discussion through meetings and many other tools in order to create the environment for this change. Because we need to change the system in this context, and it is the right time to change. Go ahead, Gina.
Yeah, so I have a question. I think in the beginning, Luca, you mentioned all the omics, and I was thinking, how can we integrate these other omics in phenomics to better understand traits in livestock? You know, so that's one question. And then Thomas, you mentioned something. You made a lot of examples regarding humans, and I was thinking, what are the best things that we can learn in human research to then apply it to animal research? And what are the challenges then to do that? Thank you.
Yeah, thank you. It is clear that the integration of phenomics with genomics is a key issue, because we need to add an additional level of annotation of the genome, but it's quite challenging. Because we are moving a little bit far from the DNA, and so the relation between variation at the DNA level and variation at the phenomic level will degrade in some way as far as we move. So it is clear that we need to add an additional level of information at the genome, because in this way we can better use the genomic information for many different purposes. And in the construction, we have a working group dedicated to this, because we need to develop novel concepts.
Yeah, when it comes to the learning, I think that the most important thing to learn is that medical researchers are not as good as they think they are. I think that, especially when it comes to the genetics side, I think that agricultural scientists can be a lot more mature, because the Hugo project was a massive effort on sequencing, and it was a huge reason for the funding was that we really hoped to find single causative genes that do things. And especially for a European population, thanks to our very tight genetic bottleneck, a lot of the more interesting diseases that we have are genetic diseases caused by a single mutation. So what have happened on the human side of research is that we think a lot about finding causative genes, and on the other hand, on the animal side, since we have now genomic selection, it means that we're actually ahead, I would say, of the human side of things when it comes to connecting phenotype to genotype. And we also have massive advantages when it comes to legislation. Because one of the reasons that I stopped working with humans and biobanking was the implementation of GDPR and how data is not allowed to move freely, which means that it becomes much, much harder to do very valuable research. And I would say that when it comes, like, for example, now when we're working on the integration between research farms in Europe, actually, I'm going to be a bit rude and say that we should definitely not let the Germans decide on how we share data. Because what happened on the biobanking side is that they got this massive focus on personal data and privacy, meaning that we're not allowed, that in some countries, like, for example, the Nordic countries are very open historically with data, meaning if I donate a sample now, then the healthcare system can keep it forever, and they can also look into my health records to see, well, of course, now I'm a male, but a classic example is miscarriages. If I was a female and donated a sample because I was pregnant and then I had a miscarriage, then that's a very valuable sample. But if you have strict regulations with consent, then you might, for example, force the person who donated the sample to renew her consent every 5 years. And the persons who are the least likely to give such a consent are the people who feel uncomfortable about being reminded of something bad happening in their history. So what have happened on the human side is that we have locked up our ability to do research. On the other side, what we can learn a lot about, I think, in the animal side, is to try to work together with the industry to figure out the different stakeholders who produce the best systems to generate data that we can use for phenotyping. We need to think about their business models to find out ways to get access to their data and not be in a problematic position. Like, for example, when I work with GigaCow, getting the data from the dairy farms is really easy because all the big manufacturers of equipment have always said the farmers own their data. So they might not help me to get the data, but they don't stop me either. So I can basically go out into the farms and collect data with the consent of the farmers. Then we have the advisory organization Vexa, who in theory, as long as the farmer consent, they're very happy to hand over the data. But they also feel that they're custodians of the data, which means that the contract for actually giving me the data is quite long and complicated. So basically, I'm not allowed to patent certain things, and I need to think about how to store the data. So it's a much longer contract. My deal with the farmers is basically, if they don't like the work we do on the SLU dairy data collection infrastructure, they stop sharing data with me, and then my career is a complete train wreck. So basically, it's the promise of mutual destruction that means that they feel comfortable giving me data. The advisor organizations was a lot more picky. And then we went to the Nordic breeding evaluation, who do all the breeding evaluation and genotyping. And they are Danish, and they have a different approach to things than Swedes quite often. So he pretty much looked at the long contract from the Swedish advisor organization. And then he was like, this is very complicated. And then his conclusion was that given how cheap it is to do genotyping in dairy, what they are afraid of is licensing costs, that someone basically paywall them from doing some kind of genetic testing. That's their fear. So the deal with them is that no one may ever patent anything based on the conclusions we make from their genetic information, because that means that they compete based on the fact that they have a lot of good technicians and service staff. So that means that for them, as long as the information is free, then they win, because they give the highest level of service for the Nordic agricultural community. So finding those, what are people willing to share and what helps them with their different business models, that is something we really need to think about for future high throughput phenotyping.
Thank you very much, Thomas. Thank you. I think we're going to wrap up this session now. Okay, we have scheduled it for an hour, so we would like to keep them in schedule. I would like to thank you both very much for joining us and for speaking to us. And I will just briefly share again the QR codes for you to join and to follow us on the social media. While I just thank you for being here, and we will be in touch for the next episode of this webinar. And well, thank you again to Luca and Thomas for their talks, interesting talks. If you, well, Gina just said it in the chat. If you have any question, please, you can write to us to this email. And see you next time.
Yeah, thank you. Thank you very much.
Thank you.
Thank you. Bye-bye.
Thank you.